Hello everyone, my name is Nalim, the Bazooka Prophet. How are you guys doing? I hope you guys are fine. I hope you guys are learning in this period. Today we'll be continuing from our previous video that we did on gluconeogenesis. And in our today's video, we'll be talking about what we call the metabolic pathway that is involved in gluconeogenesis. But please do not forget that in gluconeogenesis, we said that it is a series of steps which is also being controlled by enzymes that form glucose from non-carbohydrate substances or molecules. And examples of these non-carbohydrate molecules include the pyruvate, the lactate from glycolysis. It also includes the intermediates of the citric acid cycle, and also most carbon of the amino acids are also being used. And I give an exception that leucine and lysine are not part of it, because why? In the pathway of gluconeogenesis in animals, the first step that must be attained, in which these non-carbohydrate substances are being converted to, is the oxaloacetate. And leucine and lysine cannot, because when they are being degraded or oxidized, they form what we call the acetyl CoA, which cannot be converted to oxaloacetate in animals. But in plants, we said it can be. Acetyl CoA can be converted to glucose by a pathway called the glycosylate cycle. Now, in this pathway, we should understand some basic points about it. That in the glucogenic metabolic pathway, 11 steps are recorded, or are to be taken note of. 11 steps, and these 11 steps are controlled by enzymes. Exactly. Now, we should not forget that gluconeogenesis is a reversal of glycolysis, though they are not same. Now, gluconeogenesis and glycolysis are similar in the sense that they both share 7 enzymes. While the difference between gluconeogenesis and glycolysis in terms of their pathway is that for the 3 irreversible steps in glycolysis are bypassed in gluconeogenesis, and the irreversible steps that we have in gluconeogenesis are four steps which are controlled by enzymes. And the enzymes are, in the case of this one, it is called the pyruvate carboxylase. This one is phosphoenol pyruvate carboxy kinase. Why? It means that in these first 2 steps, they are exclusively for gluconeogenesis. Then we move to the steps in the eye, which is being catalyzed by the enzyme fructose 1,6-bisphosphatase, and lastly, that gives us the glucose, is called the glucose C phosphatase. Now, when we look at this pathway critically, we understand that from pyruvate to oxaloacetate, ATP is consumed, and CO2 enters into this pathway to give us what we call the oxaloacetate. Then from oxaloacetate to phosphoenol pyruvate, GTP is also consumed. And when this GTP is complete, please take note that this GTP is equivalent to the ATP. Take note. So that when that is done, GDP is being released, and carbon 4 oxide, which initially entered into this first step, is being given out. Take note, these 2 steps are irreversible. Now we then move to the steps that connects the gluconeogenesis with the glycolytic pathway. The first one is this one, which is from phosphoenol pyruvate to 2-phosphoglycerate. Take note, it is being controlled by an enzyme called the enolase. Enolase, as you can see, from phosphoenol pyruvate to 2-phosphoglycerate is controlled by the enzyme called what now? The enolase. While from 2-phosphoglycerate to 3-phosphoglycerate, it is controlled by the enzyme which we call the phosphoglycerate mutase. Take note, the phosphoglycerate mutase. It simply means that for exam sake or in exam condition, you can be asked, what is the enzyme that catalyzes the 2-phosphoglycerate to 3-phosphoglycerate? Take note, it is called what now? The phosphoglycerate mutase. Now, in this step, from 3-phosphoglycerate down to 1,3-bisphosphoglycerate, take note, ATP enters into the pathway, into this step. And when ATP enters, ADP is being released plus inorganic phosphate. And take note, when that happens, the enzyme that is being utilized in this step is called the phosphoglycerate kinase. Remember in our previous video, we talked about enzymes and we talked about how these enzyme names are obtained. Now we then move that from 1,3-bisphosphoglycerate straight down to these two products that are made up, which are the substrate, which are called the dihydroxyacetone phosphate and the glycerol phosphate. Please take note, from this step down to this step that we occur here, NADH enters into it, this is the reduced form, and this is what now the oxidized form. And with that is formed, what we connect this dihydroxyacetone phosphate to glycerol phosphate is what we call triose phosphate isomerase. Take note, triose phosphate isomerase. From what to isomerize, what connects these together is this enzyme. And then from here straight down to here, which we give what we call the fructose 1,6-bisphosphate. It is being controlled by the enzyme, the aldolase. Take note, aldolase. Now, we will then move to this step here, which is fructose 1,6-bisphosphate. We will now result in the removal of the phosphate group, which will now give us the last product called what? The glucose. Now, please take note. In this step, which is another important step, in fact, in gluconeogenesis, the rate limiting step of gluconeogenesis is this step that you are seeing here. That from fructose 1,6-bisphosphate straight down to fructose 6-phosphate, in which there's an entrance of one molecule of water to release one inorganic phosphate. The enzyme that controls this step is called the fructose 1,6-bisphosphatase. Now take note, the rate limiting step or the rate limiting enzyme is this one, which is called the fructose 1,6-bisphosphatase. Now please do not forget that from pyruvate to oxaloacetate, they must occur in the mitochondrial matrix only. While from oxaloacetate down to phosphoenol pyruvate, this one occur in both the mitochondrial matrix and the cytosol. But in this other irreversible step, this one occur only in the cytosol, and in the last irreversible step, this one now occur in the endoplasmic reticulum. Now take note that from fructose 6-phosphate to glucose 6-phosphate, this step is also what connects the glycolytic pathway with the gluconeogenetic pathway, and it is controlled by the enzyme called phosphoglucose isomerase. And lastly, from glucose 6-phosphate to give me glucose, the enzyme involved is called the glucose 6 phosphatase. So it means that from glucose 6 phosphate to give me glucose, the enzyme that will catalyze that reaction is called the glucose 6 phosphatase. And on that very note, we understand the 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 summary behind it, that first of all 11 steps are involved, and that secondly the 4 irreversible steps which are involved only in the gluconeogenesis are A, and you have it, which is the pyruvate carboxylase; B, the phosphoenol pyruvate carboxy kinase. Then we move to the I, which is called the fructose 1,6 phosphatase, and lastly, which is K, is called glucose 6 phosphatase. And on that very note, we come to the end of this block of lecture on the glucogenic metabolic pathway. And remember yourself, and adrenaline, the Bazooka Prophet. God bless you.